So we talked a bit about thinking about T cell development and the thymus last time, but there was one set of experiments that I didn't have a chance to completely tell you about last time that I wanted to talk through. This set of experiments involves the use of TCR transgenic mice, which I told you about last time as well. So with these mice, we take one T cell from the mouse. It recognizes one antigen on either class 1 or class 2. Here, it's an influenza-specific antigen on class 1. And we take the alpha and beta T cell receptor genes from that T cell and put them into our mouse as transgenes. This will result in allelic exclusion, so that 100% of the T cells in our mouse that develop will become sort of clones of that original T cell. And so you can see we'll have our developing double negatives, we'll have some developing double positives, and in this case, because we started out with the CD8, we'll get only CD8 single positives, no CD4 single positives, and when we look in the periphery, we'll only see CD8 positive T cells. This mouse would not be particularly happy or healthy because of this T cell lack. It would only survive in the nice, clean lab environment, but experimentally, it would be interesting for looking at T cell development. And very frequently, when people are trying to do T cell experiments, they make one of these mice so that they can have a good source of T cells specific to one particular antigen. And so these are sort of important for doing some T cell biology work as well. I first told you a little bit about one way that people can do these experiments, where we might say take a T cell clone from a mouse that is on the H2K background, clone those alpha and beta TCR genes, and either put them into an H2K mouse or an H2D mouse. And really, you're putting it into the embryo and letting the mouse develop from there. And if you put it into one with matching MHC, you get some developing T cells, and they successfully develop into those CD8s. We see exactly what we saw on the previous slide. If we, however, were to insert those alpha beta TCR genes into an H2D mouse background, as the T cells are developing, they will be trying really hard to develop. We'll see some double positives in the thymus, but we will never see our single positives that have finished maturing because they will not see self in that thymus, and they will all die by neglect. Because the thymus is going to be H2D, these T cells are all going to be trying to recognize H2K. They're going to not recognize, and they're all going to die. I will also tell you that on the practice problems, there is a question that has to do with these types of things that I think is the most fascinating. Like, I think it's such a good question. I also think it's kind of hard and unfair, and so I would never actually ask it on an exam, but I will tell you that if you work through the process of trying to understand that question, you will understand this stuff really well. I love the question, but totally can't ask it, and it always kind of makes me sad. So I would highly recommend going through some of those practice problems. So these types of experiments where the alpha and beta TCR genes were put into mice of different MHC backgrounds were part of the way that we figured out sort of the order that different cells in the thymus developed. But the other way that we figured out a lot of this was in doing one other set of experiments. And in that set of experiments, it was really starting out as the same thing. So again, we have a T cell that is specific for an antigen. And we are cloning out the alpha and beta T cell receptor genes and putting them into embryos. Same exact thing. The difference here is that we are picking an antigen that is called HY. HY is an antigen that is encoded by the Y chromosome. So in females, this is a foreign antigen, whereas in males, this is a self-antigen. And we can examine the development of HY-specific T cells in both females and in males. That lets us show whether HY-specific T cells can develop. You kind of have a nice control where you can say, hey, look, they develop normally in females when they're not self. It's not like HY is like some wacky antigen. But then you can also compare in males where we see this is a self antigen. So the HY antigen in this case is presented by H2D of B. So we can take the alpha and beta T cell receptors from this T cell and put it into a mouse that is of the H2B type. That means one of its MHC class 1 molecules is D of B. Since this mouse is a female, it does not have HY, so HY is a non-self antigen. And in this mouse, we got some double negatives developing, we got some double positives developing, and we get a whole lot of CD8s. This is actually showing you the real data from the paper, and so it says you got some CD4s, which you did. They actually did this experiment before they knew that TCR alpha was bad at allelic exclusion. And so if you look at this actual data of this experiment, it's a little messy. We now would do this experiment and make these mice also RAG deficient, so we could see absolutely no other rearrangement, and it would be entirely CD8s. When they did the same experiment where they put the alpha and beta genes into the female of H2D background, it has D of D as one of its MHCs. It doesn't have D of B because it's the D kind. HY is still not there because this is still a female. We have cells that are attempting to develop as double negatives, cells that start developing as double positives, but we see no single positives. This is actually the exact same scenario as what you saw with the influenza before, because in both cases it's a non-self with matched or non-matched MHC. And we can see the same data here. So this is basically what the thymus looks like of their wild-type mice. If we look at, this is also their lovely messy data, they should have gotten only CD8s. So double negs, double positives, CD8s. They did get a few double CD4s because it was messy. You can see they also did some mice that had different MHCs. As long as they got this one, they're good. So you can see the same thing. But if they look at mice that only have H2D, so this is this is an antigen that is not recognized by MHC. You can see that they have double negatives, they have double positives, no single positives, because those cells are dying by neglect. Yep. Okay. And so the thing that becomes interesting in this experiment is that you can then look at the males who are H2B, where this is a self antigen, as opposed to the females where this is a non-self antigen. And you can start to understand that development. And so they did the exact same experiment, the same alpha and beta T cell receptor, but they put them into male embryos that were of the H2B type. Here, HY is a self antigen because these guys have Y chromosomes. And what we end up seeing, so here's the female and here's the male, where really we have double negatives. You can see that it kind of looks like there's other stuff, but really they aren't full populations. It's just they kind of tried to express things and they're a mess. And so in fact, we have just double negatives and we don't even really get any double positives because all of those double positives are being killed as self-reactive cells. And so this helped further our understanding that double negatives came first, double positives came second, and then single positives came later. And so the combination of these types of experiments is really how we figured out this process of T cell development in the thymus, is by looking at sort of combinations of what happens in all of these types of mice. So at this point, we finally get to go to the periphery. We have developed our B cells, we have developed our T cells, and finally we can actually have those cells leave and start to see antigen. So we'll have our B cells leaving the bone marrow and moving into blood flow, and we'll have our T cells leaving the thymus and moving into the blood flow. And And now they are going to be circulating throughout the body. So some of them will end up in barrier organs. Others will end up in lymph nodes or the spleen. Some will be moving through the lymphatic vessels, and they will be circulating around. And these secondary lymphoid organs are the places where our B cells and our T cells are finally going to meet antigen and start to make a response. And so this is, as I've heard other people say, this is where the party happens. One thing we can think a little bit about is where are the lymphocytes once they leave the primary lymphoid organs. These are two different views of this from two different versions of your textbook, and they both have kind of strengths and weaknesses. So one thing that you can see is that it shows on the left the lymphocytes circuiting in the blood. And if a lymphocyte was going to make a full circuit from heart all the way through the blood back to the heart, it would take 30 minutes. So it can just basically circle every 30 minutes. Some will leave and go to mucosal tissues. Some will leave and go to spleen. Some will leave and go to lymph nodes. Some will actually then come back to the blood. Some will go back to the lymph. One thing that you might notice is that these percents do not add up to 100% at all. And so I would not take them as great answers of exactly where the cells are, and it can be somewhat difficult for us to know where the cells are because some of these locations are easier to measure than others. And so our exact understanding of where those cells are can be a bit biased. This view from the more updated version of your textbook says 10% go to barrier organs, 40% go to lymph nodes, 50% go to the spleen. I think those numbers are super sketch. But in the end, you can see that these lymphocytes are going to a few different locations. Today, we are really going to be talking about the time that they spend in the secondary lymphoid organs, either the lymph node or the spleen, which according to this version is 90% of the cells. Here is a decent percent of the cells as well. And so the cells will be leaving the blood or leaving the lymph circulation and spending some time in a secondary lymphoid organ, maybe 5 hours in the case of the spleen or 12 hours in the case of a lymph node, looking for antigen, ideally so that they can become activated. We're going to sort of assume that this secondary lymphoid organ is a lymph node just for sort of simplicity's sake. Though it could be any secondary lymphoid organ. To me, I think of the spleen as a really big lymph node. And so I'm going to talk about the lymph node, but this could be any secondary lymphoid organ. And if we look at the structure of that secondary lymphoid organ, particularly a lymph node, there are a few important things to notice. So first, you can see that there are blood vessels going both in and out. So we've got a vein and an artery coming into that lymph node. When we dissected the mouse and you saw the lymph nodes, you saw that there were vessels, there was vasculature coming in. We also have incoming and outgoing lymphatic vessels to allow transport of things in and out through the lymphatic part of the circulatory system, through the lymphatic system. There are then specialized structures within that secondary lymphoid organ. Some of them are going to be B cell zones, some are T cell zones, and there are other specific types of structures where we're going to see our cells moving. One place that I am going to specifically mention has to do with the vasculature. When we have these vessels that are in the lymph node, often they are specialized to look like this, and they are known as high endothelial venules, or HEVs. And so we're going to talk at some points about what happens in the HEV, the high endothelial venule. I didn't always get a couple things about the high endothelial venule, so just so you know, the reason why it's called the high endothelial venule is because these cells, these are endothelial cells on the border, are tall. They're high. So it has high endothelium. And so for the longest time, I couldn't figure out where in the world they were getting that name. It's because these cells are tall. And so we will see the HEV, or the high endothelial venule, being important here. We're going to see versions of this slide multiple times throughout the next few class periods. They all get to this question about adaptive immunity. We've learned about innate immunity versus adaptive before. And one of the key things that we talked about with adaptive immunity is that after we have infection, it takes a little while before we have a good adaptive immune response. So here you can see, our antibody or T-cell response on the upper left taking sort of time, weeks. Everybody always wants numbers. And usually if you look at these figures, they're very vague in the numbers. The reason is because actually it depends on the pathogen and the dose and things like that. But everyone wants real numbers. So this is a paper that I published where I actually measured the response to one particular antigen over time. And you can see that in my experiment, at day 14, I had the maximum adaptive immune response. I couldn't measure anything at day 4, could start to measure at day 7, could measure pretty well at day 10. So there was sort of this lag period from when I infected my mice. And the question that we're going to be addressing partially today, and in part some of the other upcoming days, is what the heck takes so long? What is the adaptive immune system doing that's going to take all of this time? What's going on in those days? These things that are listed here are some of the different pieces of this. So one of the big things that's going to happen is we're going to have things like antigen making its way to the secondary lymphoid organ, as well as B cells and T cells getting to the secondary lymphoid organ. So the first thing that has to happen is travel. After that traveling happens, we're going to have to talk about things like how recognition happens, how the cells are activated, how they proliferate, how they change. But travel is going to be a big part of it. And in fact, if you look here near the end, then the T cells have to travel again to get from the lymphoid organ to the site of infection. So in the case of the flu, they'd have to get from the lymph node back to the lung. And so today we're going to be talking about the travel parts. That travel could be this beginning travel, that travel could be this ending travel. But we're really going to work through what's happening there, because we can't see any of the other pieces of this response happening until everything has traveled to the right location. This process is usually referred to as the process of trafficking. And so trafficking is our first answer of what is taking so long. And so this sort of shows you a very broad overview that for T cells, a few hours after infection, APCs might be sort of waking up. It's going to take almost 24 hours to get your antigen into that lymph node. It might take about then another 24 hours for T cells and antigen-presenting cells to actually meet up and do something. Then the T cell is going to kind of activate for 4 days, and then it'll start leaving. Similarly with B cells, we've got to wait for antigen to get there. We've got to get B cells to get there. B cells have to move around a lot. And so there are all these travel steps that are going to be sort of tricky. The first thing that we've got to get into our secondary lymphoid organ is our antigen. And it turns out getting antigen into this secondary lymphoid organ is the easy part. So we will have some antigen. For example, it might be the red dot antigen that you acquired when you stepped on a rusty nail that will make its way into the tissues. There are all sorts of open lymphatic vessels throughout the body that are trying to pick up liquid, as we talked about earlier in the semester, that will allow those antigens in as we have body movements that will push those antigens straight through into the draining lymph node that drains that particular region of the body. Sometimes there might be a phagocytic cell, like a dendritic cell, that carries the antigen. Sometimes the antigen is carried as, it just goes as free antigen. And so the antigen can move in either way. You can see that here. You can also see that here where there are some nice open lymphoid vessels where things can move either on a cell or free to get into the regional secondary lymph.
This is another of the files where the rest of the original audio is completely obscured by static. So I am re-recording the rest of this lecture at a later date. I apologize if it doesn't match your notes perfectly, and I apologize that we don't have the student interactions, but hopefully you'll be able to actually hear what I'm saying in this situation. I left off talking about this slide where we saw antigens being transferred to lymph nodes through the draining lymph or with some type of antigen presenting cell that may phagocytose those antigens and travel to the lymph node. There are lymph nodes that drain different regions of the body that will collect antigens and antigen presenting cells that have been found in that region. This way, your adaptive immune cells don't need to go through all parts of the body. They can simply go from lymph node to lymph node, looking to see if their antigen is in that particular body region, and then we'll need to only look at the small region that is drained by that particular lymph node. On these two slides, you can see examples of lymphatics as well as the large array of draining lymph nodes that exist in order to drain different parts of the body. We were just talking about how antigens can make their way into the lymph node. We also, of course, need T cells to make their way into the lymph node if we want to have some type of response. There are two ways that naive T cells can make their way into the lymph node. One is by traveling through blood vessels that will enter into the lymph node. When you remember our mouse lab, you remember that we could see blood vessels going in and out of the lymph node, and T cells are able to use those blood vessels to enter the lymph node. There are specific special types of vessels, blood vessels, that go through lymph nodes called high endothelial venules, or HEVs, that T cells often will use to actually leave the blood vessel and enter the lymph node. We'll talk a little bit more about the HEV later. I was originally confused when I learned about HEVs because I didn't get why they had the name high endothelial venule. It turns out that their cells are very tall, which is why they are called high endothelial venules. T cells that have left the blood can also travel through lymphatics in order to travel from lymph node to lymph node and not try to make the circuit of the entire body through the blood. The process that I'm going to tell you about is the process that is known as leukocyte trafficking. This is the process that, as you see in the middle, naive T cells or naive B cells can use to enter into the lymph node through the high endothelial venule, through the blood vessel. However, it's the same set of steps that we will learn today that other types of leukocytes will use in order to leave blood vessels to go to other locations in the body. So this is the same process that neutrophils or monocytes could use if they want to leave the blood vessel to go to a site of infection or a site of inflammation. This was actually discussed a bit in your textbook in the innate immune responses chapter. However, since the steps are the same steps, I decided to only talk through them once although it's really the same process here. Also, after our T cell has been activated and it wants to go to a specific body location in the region that it should go to based on which draining lymph node it was activated in, and actually find and deal with infection. So perhaps this might be a T cell that was activated and now actually wants to go to the lung and do something about flu infected cells. That T cell will again travel through blood vessels and will actually go to the infected tissue. And the process that we're going to see today, the same set of steps will be used for that cell to migrate into the site of infection. So while we see this example today first for naive T cells going to the secondary lymphoid organ, the same general processes can be used in a lot of situations throughout the immune system. One thing that is very interesting about this process is that we have a really good technique that we can use to study it called intravital microscopy. In intravital microscopy, we can actually perform microscopy on cells in organs in live animals. Typically, what you will see is you will see a situation where the popliteal lymph node, which is the lymph node in the groin, is surgically exposed. That one is chosen because it's very close under the skin, so we don't have to disturb much tissue in order to make that lymph node visible and outside of the skin. And then a microscope is used to look directly into that lymph node. The mouse is, of course, anesthetized so as not to move. Sometimes we can look at the cells that are in that mouse normally. Other times we might transfer in antigen presenting cells and or T cells, as is shown on the left here. Oftentimes we do that because those cells might be labeled with some type of fluorescent molecule that will make them easier to see under the microscope. So what we will tend to do is have, as you can see on the right-hand side, a microscope sort of looking directly into that exposed lymph node of our mouse, and we'll be able to watch cells moving. Our mouse is anesthetized, usually through something coming in through a tail vein, as well as immobilized. It has a heat pad, but we're able to watch cells moving through the lymph node or through vessels in real time in that mouse. And so this has really allowed us to understand quite a bit about this trafficking process. We should think a little bit about some of the details of blood flow and what types of problems our cells might encounter. One of the major problems that we can think about for our cells is the way that blood flow is moving through these small vessels and how quickly, relative to the size of the cells, they are moving. These cells are moving incredibly, incredibly quickly. There is, in fact, as much shear stress on them as you might see in some very high-level whitewater rapids. And so movement out of the blood flow can be a real problem for white blood cells. And you can kind of think about this in the same way, or think of the same types of problems that you might have when driving on a highway here. You can imagine that when driving on a highway and you're going really fast, you don't want to just randomly stop and say that, oh, look, it's my exit. Stop in the middle of the flow of traffic and then try to go to the side to your exit. You would realize that quite quickly you would be hit by lots of other oncoming cars. It would probably be difficult to stop right away. This would not work out quite nicely. And so instead, of course, you do things like put on a signal, you slow down, you begin to move into the right-hand lane out of the flow of fast traffic, and go through sort of this sequential process of getting ready to exit. Similarly, a blood cell, when it wants to exit the very fast moving stream of blood flow, needs to do a similar process. I'm going to show you the video from this link for a few minutes, for just a second, and hopefully it will help you appreciate just how fast the individual cells are moving. Particularly think about this from the cell's perspective, given how small it is, how fast it's moving at the length of a cell body during this process of blood flow. So here in the middle, you can see all sorts of blood cells moving very quickly in order to go through the vessels. You can see some capillaries and things like that. And so what I hope you appreciate is just how fast these cells are moving and how potentially problematic it would be for a cell to simply just stop in this process. As a result, our cell is going to do multiple actions, go through multiple steps in order to leave the blood vessel sort of safely, just like you would go through multiple steps in order to leave the highway safely. The first of those steps is a step that's called rolling. Then we'll see a step called activation. Sometimes rolling is called sticking. Sometimes activation is talked about as chemokine signaling. We can then see a final step, which is sometimes known as arrest or firm arrest. Some people call it adhesion, some people call it stopping. And we can then see a final step called diapedesis. I'm mostly going to spend my time talking about the first three steps as we understand a bit more about some of the details that have to happen for those three steps, and I'll only briefly talk about diapedesis at the end. Just like in the car example, with rolling, this cell is going to be attracted to the wall of the vessel. So it's going to move out of the middle of the vessel with fast flow and move toward the edge, as well as starting to slow down. That's really what we see with the process of rolling. The cell isn't going to come to an immediate stop, but it's going to slow down. It's going to start rolling instead of going fast and move to the side of the vessel. And you can see that in this video.
Leukocytes are white blood cells that help fight infection. At sites of injury, infection, or inflammation, cytokines are released and stimulate endothelial cells that line adjacent blood vessels. The endothelial cells then express surface proteins called selectins. Selectins bind to carbohydrates displayed on the membrane of the leukocytes, causing them to stick to the walls of the blood vessels. This binding interaction is of sufficiently low affinity that the leukocytes can literally roll along the vessel walls in search for points to exit the vessel. There, they adhere tightly and squeeze between endothelial cells without disrupting the vessel walls, then crawl out of the blood vessel into the adjacent connective tissue. Here, leukocyte rolling is observed directly in an anesthetized mouse.
Notice
the up and down movement of the frame is due to the mouse's breathing.
Notice the change in speed of that rolling cell
Two blood
compared
vessels
to
are
all
shown.
of the fast
The upper
cells
one is an
moving,
artery with blood
around
flowing
it, where
from
you
right
can't
to
make
left.
out those cells.
The lower one is a vein with blood flowing from left to right. Leukocytes only adhere to the surface of veins. They do not crawl out of arteries. Some leukocytes are firmly attached and are in the process of crawling through the vessel walls, whereas others have already left the vessel and are seen in the surrounding connective tissue. When the blood flow is stopped temporarily by gently clamping the vessels, we can appreciate how densely both vessels are filled with red blood cells. Red blood cells do not interact with the vessel walls and move so fast under normal flow that we cannot see them. When the blood flow is restored, some of the leukocytes continue rolling, whereas all non-interacting cells are immediately washed away by the shear.
So there you were able to see how our cells move to the outside of the vessel wall and start to move more slowly compared to the fast main stream of the blood flow. The cells are able to move over to the side of the vessel wall and start this rolling process because of interactions between a type of protein called a selectin and its ligand. A selectin was named so because it is a type of protein called a lectin, and lectins are carbohydrate binding proteins. So our selectin will bind to some type of carbohydrate ligand that will slow down our cell and have it be attracted to the side of the wall. The interaction between selectins and their carbohydrate ligands is of relatively low affinity, and so the interaction can attach but then come off quite easily. You can almost think of this as being somewhat like Velcro, and so the cell can sort of still be moving as it's pushed by the sort of physical pace of blood flow, but be slowed down as it keeps catching and recatching on the side of the wall. There are three types of selectins. They are known as L-selectin, P-selectin, and E-selectin. They are also known as CD62L, CD62P, and CD62E. L-selectin is often found on some lymphocytes and leukocytes. P-selectin is found on platelets. E-selectin is found on the endothelium. And these bind to some type of carbohydrate ligand or CAM. I'm not specifying right now which one is on the cell and which is on the vessel wall, because during the different situations that I've mentioned before, either the neutrophil trafficking, the naive T-cell trafficking, the activated T-cell trafficking, for example, the location of the selectin and the location of the carbohydrate will differ, and I will talk about that a little bit at the end. And so here you can see this process of rolling where we have the carbohydrate that is going to bind to a selectin. It's going to bind weakly, and so that cell is still going to be getting pushed by blood flow, but you can see as it's getting pushed, it keeps re-catching on another selectin and eventually sort of slowing down to roll along that vessel wall. And then you can see that process of rolling in this next short video.
While red blood cells are carried away at high velocity by a strong blood flow, leukocytes roll slowly on endothelial cells. P-selectins on endothelial cells interact with PSGL1, a glycoprotein on leukocyte microvilli. Leukocytes, pushed by the blood flow, adhere and roll on endothelial cells because existing interactions are broken, while new ones are formed. These interactions are possible because the extended extracellular domains of both proteins emerge from the extracellular matrix, which covers the surface of both cell types.
Once the cell has begun rolling, it's ready for the next step to happen. I'm going to say a few things about that next step, but I want to take a second to think a little bit about the goal, which is the third step. Eventually, we need our cell to actually stop and be able to crawl through the vessel wall. You can imagine that in order to stop, our cell is going to need to have some type of very strong interaction between some protein and its ligand that's able to immediately stop the cell. You can also imagine that it might be dangerous for our cell to have this sort of protein with a strong interaction be present at all times. The cell could accidentally stop at the wrong location. Perhaps it could stop without rolling first, at which point that cell would get destroyed by contact with sort of the oncoming shear flow. And so the stopping proteins that we're going to see are not being made by the cell at all times. Those or are not ready to stop the cell at all times. They have to be activated. And so we need to see a signaling event that happens in our cell in order to turn on the stopping proteins. This activation step is the step that we're going to see next. In addition, we're going to see interactions between a protein and its ligand here. And with our 3 steps of rolling, activation, and arrest, we can also have very specific interactions between proteins and ligands that are required. So that this can help to make this process selective and allow a particular type of cell to go to a particular location. You can sort of think of this as being like an area code where 919 we'll call North Carolina and 973 we'll call New Jersey. Similarly, one type of selectin will lead to rolling, but then it requires another specific interaction to make activation happen and yet another to make arrest happen. So that makes this process very specific. A cell, for example, could start rolling and it won't keep going through this process if it's in the wrong location and doesn't get the next type of signal. That signal happens through proteins that are known as chemokines binding to their receptors. Chemokines, as we learned about earlier in the semester, are chemotactic cytokines. They're cytokines that are involved in cell movement. Chemokines bind to chemokine receptors, one of which is shown here, although it is shown incorrectly, because a chemokine receptor will have 7 transmembrane domains and will be coupled to a G protein. It is, of course, known as a G protein coupled receptor, which will lead to a number of signaling events in the cell. In the end, those signaling events will all allow for things like chemotaxis because of things like cytoskeletal changes in the cell, as well as turning on that stopping protein, as I mentioned. When we see chemokines, we often see them named as things like CXCL and then a number, or CCL and a number. The L means ligand. It means we're talking about the chemokine. The CXC or the CC tells us a little bit about the structure of the chemokine as is shown here. If we think about chemokine receptors, the names are things like CXCR and a number, or CCR and then a number, R standing for receptor. Unfortunately, CCL1 does not necessarily bind CCR1, but this nomenclature should help you recognize when you are seeing a chemokine or a chemokine receptor. Each leukocyte has a unique set of chemokine receptors. Again, this allows for sort of that area code type of model where each of these cell types could move to different locations in the body in different types of situations. And we can see changes in chemokines on a cell between a resting cell and an activated cell, for example, as those cells likely have different needs in terms of where they might want to travel. So we're going to see some signaling through those chemokine receptors, and those chemokine receptors will then allow us to do the 3rd step of this process, the arrest or adhesion step. Firm arrest or adhesion is again mediated by interactions by some specific proteins. In this case, the interaction is between a type of protein called an integrin, which will interact with some type of cell adhesion molecule or CAM. Integrins have two chains, an alpha chain and a beta chain. And the integrin will always be on the same cell type as the chemokine receptor from the previous step, because the integrin is going to react to signals that came through the chemokine receptor. And the binding between integrins and their associated CAMs is extremely strong and will actually stop our cell. Typically, we name integrins as something like alpha 4 beta 1, which tell us which integrin alpha chain and which integrin beta chain is being used. Sometimes there are common names for some of these integrins. So for example, alpha M beta 2 is often referred to as MAC1. But typically if you see something named alpha number, beta number, that is going to be some type of integrin. And there are many different types of CAMs or cell adhesion molecules that are our integrin ligands. Integrins, as I mentioned before, need to be activated. They have this very strong interaction with their CAM ligand and are not going to be active all the time because we don't want our cell to be stopping randomly. So normally our integrins are present in what is known as this bent conformation. You can see it on the left of both versions of this figure, where the integrins are sort of folded back on themselves and their binding site is sort of hidden. Sometimes this is known as a jackknife conformation. When there is signaling through a chemokine receptor, that will lead to some changes in the integrin, and the integrin will actually extend so that its binding site is now available, and it will be able to interact with its corresponding CAM. This will be a high affinity interaction that will completely bring our cell to a stop. And you can see all of this process in this part of the video.
Fuses with the plasma membrane. Proteins contained in the vesicle's lumen are secreted, and proteins embedded in the vesicle's membrane diffuse in the cell membrane. At sites of inflammation, chemokines secreted by endothelial cells bind to the extracellular domains of G-protein-coupled membrane receptors. This binding causes a conformational change in the cytosolic portion of the receptor, and the consequent activation of a subunit of the G protein. The activation of the G protein subunit triggers a cascade of protein activation, which in turn leads to the activation and clustering of integrins inside lipid rafts. A dramatic conformational change occurs in the extracellular domain of the activated integrins. This now allows for their interaction with ICAM proteins displayed at the surface of the endothelial cells. These strong interactions immobilize the rolling leukocyte at the site of inflammation. Additional signaling events cause a profound reorganization of the cytoskeleton, resulting in the spreading of one edge of the leukocyte. The leading edge of the leukocyte inserts itself between endothelial cells, and the leukocyte migrates through the blood vessel wall into the inflamed tissue. Rolling, activation, adhesion, and transendothelial migration are the 4 steps of a process called leukocyte extravasation.
And so now we've seen the first 3 steps of this process: the rolling step, where the cell slows down and starts to stick to the wall, the activation step through chemokine receptor interaction, and the firm arrest, where the cell actually stops and is ready to leave. The final step is known either as diapedesis, extravasation, or transendothelial migration. At this step, our leukocyte is going to have some major changes in its shape via the cytoskeleton, which will allow it to actually crawl in between endothelial cell walls in the vessel. Much less is known about the details of this process, although we do know that this process requires quite a bit of chemokine activity as well. As I mentioned before, these same steps are used whether we're talking about a neutrophil moving to a site of inflammation, a naive T or B cell moving to a site of a secondary lymph node so it can find antigen, or an activated T cell moving into a site of infection. And so we can think about the specific examples of what happens in each of these cases, although again, we're going to see the same steps of the process. When we see neutrophils moving to the site of inflammation, oftentimes what we will see is either P or E selectin. The P or E selectin will be on the surface of the endothelial cell, and neutrophils will have a carbohydrate ligand on their surface. The carbohydrate ligand is always on the surface of the neutrophil. That neutrophil is always a neutrophil. It always wants to try to go to a site of inflammation. Sites of inflammation are not always inflamed. Sometimes they might be your regular skin. And so your regular skin doesn't have P-selectin and E-selectin on the surface of its cells because it doesn't normally need to call in neutrophils. Only when we start an inflammatory response will those endothelial cells start to make P and E selectin to start to call in those neutrophils. In addition, we will see the site of inflammation, in my example before, the skin, start making a chemokine. The chemokine in this case is somewhat poorly named. It's called IL-8. And so we will see the induction of IL-8 again to try to call in the neutrophils because the site has changed and now is a site of inflammation where we want to call in neutrophils. Neutrophils will always have the receptor for IL-8 on their surface because they're always listening for the call. We will then see some integrins like LFA1 or MAC1. I'll show them again on the next slide. Those will be on the neutrophil. They will become activated when the neutrophil responds to the IL-8 chemokine because the neutrophil will have the chemokine receptor, and this will allow those neutrophils to stop. You can see that same process here. As our endothelium gets inflamed, it will start making different selectins, particularly P and E selectin. Those will bind to a carbohydrate ligand on a neutrophil as it happens to come by. We'll also see chemokines, in particular IL-8, coming from the inflamed endothelium, which will interact with the chemokine receptor on the surface of the neutrophil. This will turn on the activation of our integrins, which will then bind to their CAM partners. Alternatively, we can think about how a naive T cell or B cell might move to a secondary lymphoid organ. This process will start with the naive T cell expressing L-selectin, CD62L. A lymphocyte turns on CD62L when it is naive, when it leaves the primary lymphoid organ. So that's the one that's turned on and off. A high endothelial venule is always a high endothelial venule. It always wants to call in naive lymphocytes, and so it will always have the carbohydrate ligand that binds to L-selectin. The L-selectin is the thing that's turned on or off here. The lymphocyte will also have a chemokine receptor called CCR7 on its surface. Again, that is turned on and off. It's turned on when a cell is naive, a T cell or B cell is naive. And the high endothelial venule will always be making the chemokines CCL21 and CCL19, which bind to the chemokine receptor CCR7, because the high endothelial venule is always trying to call in naive T or B cells. Then we see the integrin LFA1, also known as alpha 4 beta 1, which is going to bind to an integrin ligand, a CAM, in order to attract those T or B cells, allow them to stop at the high endothelial venule. The CAM will be made by the high endothelial venule, and the integrin will be made by the naive lymphocyte. And so here you can see in red are naive T cells, which can leave blood vessels through the high endothelial venule and will then go to lymph nodes. If they don't find their antigen, they can then keep moving into other lymph nodes, and they will keep their CD62L, their L-selectin, because they still want to be able to access that high endothelial venule. They will keep their CCR7 because they still want, again, to access the high endothelial venule. However, if that cell becomes activated, that cell no longer wants to travel from lymph node to lymph node. That cell now wants to leave the lymph node and go into the peripheral tissue where it can actually find and combat its antigen. You might, as I mentioned before, imagine a T cell that responds to the flu that now needs to leave the lymph node and go to the lung in order to actually combat that flu infection. Now our T cell doesn't want to go to the lymph node anymore. So our T cell is going to turn off production of CD62L L-selectin. So it no longer is attracted to high endothelial venules. It might now be doing some different types of selectin interactions. Our cell is also going to turn off CCR7 because, again, the cell no longer wants to be attracted to the high endothelial venule. It wants to be going out to peripheral tissues. And so the cell will have some different chemokine receptors. Exactly what those are will depend on which type of tissue that activated T cell is going to, sort of which area code that T cell needs to have to go to its particular body site to deal with infection. And so here you can see the high endothelial venule, which we've been talking about. You can notice that the cells of the wall of the high endothelial venule are quite tall. They are high endothelium. And so the high endothelial venule is always making the carbohydrate ligand for L-selectin because it always wants to attract naive T cells or B cells, as well as always making the chemokines CCL19 and CCL21, which can attract those naive T cells or B cells as well. And again, you can see that same process here of the naive lymphocyte trafficking to lymph nodes. We first can see L-selectin binding to its carbohydrate ligand. It's shown here as CD34. Once L-selectin binds to its carbohydrate ligand, that cell will start to do rolling. We can then see CCR7, which is on the T cell, interact with CL21 and CCL19, the chemokines. Then we'll see LFA1, the integrin that is activated. It will bind its partner, ICAM1, and then the lymphocyte will leave the blood and enter the lymph node. All of this, of course, is happening at the high endothelial venule in the case of a naive lymphocyte trying to go to a lymph node. Once our naive lymphocyte has left the high endothelial venule and gone into the secondary lymphoid organ, it is going to start to make some type of response. And that's what we're going to be talking about in some of the next class periods. However, if you actually look at a cross-section of some of those secondary lymphoid organs, you will notice that there are a lot of different sections within those secondary lymphoid organs. There are zones that are known as T cell zones and B cell zones, and all sorts of different compartments within those secondary lymphoid organs. Those are established because different parts of the secondary lymphoid organ make different types of chemokines, and so they call or recruit different cell types to move there. And so once the cell has left the high endothelial venule, it will follow a chemokine trail that will help it go to its particular location in the secondary lymphoid organ. And then that cell will be ready to go to start its activation, which is what we will talk about in the next class.